It's 5:10, just on time. We will move on to the presentation by Devin Flawd and Tom Chance from the Lummi Nation Natural Resources Department. They will be presenting on salmonid research, monitoring, and restoration efforts in the South Fork of the Nooksack River. So it should be pretty interesting. And if our presenters have any trouble sharing their screen or anything like that, please let us know.
Thanks. I am going to try and get that up right now. And just a heads up, I'm going to be the only presenter tonight. So yes.
So Devin, I just made you co-host, so you should be able to share your screen fairly easily.
Okay. Can everybody see that?
Yes, looks great.
Excellent. Alan, can you see that all right?
Yes.
All right, great. Yes, thanks everyone for tuning in. Today I'm going to be talking a bit about research, monitoring, and restoration efforts in the South Fork. Just a few notes before we dive in. There's a lot to cover, much more than I think I can reasonably cover, in one session here. So I'm going to be providing sort of a high-level overview on different aspects of our monitoring program, restoration efforts, what the goals are, hatchery operations, and so I hope to have some time at the end that we can have, an open discussion if folks have any questions. But I hope to leave everyone with a little bit more information on kind of how things are organized and how we're carrying out and what the goals are and how we're going about tackling them in the long run. So just an organizational outline, talk about program organization and goals. Then a brief overview of status and trends. There was sort of an interest in multiple species and how they're doing in the South Fork. So I'm going to be focusing mostly on Chinook, but also we'll give a little bit of information on some of the other species that we're tracking. Talk about our South Fork Chinook, Nooksack Chinook rescue program, the hatchery operations, our monitoring and evaluation, and then some of the habitat restoration actions that have been going on. So program organization. So the Nooksack co-managers are responsible for, specifically tracking the status and trends of the Nooksack populations, Chinook, Chum, Coho, any populations that are present in the basin. We work together. I think that's important to keep in the back of your minds, just because a few of the things that I'll be talking about, I do focus a little bit more on the Lummi side of things, but just keep in mind that I'm not necessarily being informative on all the efforts that are going on from the Nooksack tribe or WDFW. Within our natural resources department, we have three main programs that operate that I'm going to speak about tonight. We also have, harvest program and water quality, water monitoring program that I'm not going to speak a whole lot about. But within these programs, our stock assessment program, we are focusing on population research and monitoring. Our salmon enhancement program runs the Skookum hatchery in the South Fork. And then our watershed restoration program is conducting habitat restoration and working on those project implementation. And for our stock assessment program, we're concerned with providing the technical data and information used in the regulation of fish populations. I'll talk a little bit more about that, about specifics. The salmon enhancement program is there to promote hatchery production and maintain harvest opportunities and really conserve the population. I say mitigate population loss, but it's really more about conserving the population after long-term population losses. And the watershed restoration program is focusing on preserving and restoring habitat to properly functioning conditions. So I think it'll be really helpful too, as we get in, to provide some definitions for some terms that I'm going to be using. I noticed that we talk a lot about these things related to salmon, but I've also found that you find slightly different variations of some common terms that we all use. So I want to make sure we're all on the same page. So when I talk about escapement, that I'll use a couple of times, that refers to the number of fish that have avoided or escaped other sources of mortality that leads to spawning. That would include any natural mortality that occurs throughout the life cycle. That would include harvest fisheries that they have avoided, and then ultimately whether or not they, are able to successfully migrate up to the spawning grounds and successfully spawn. So hatchery origin, fish that are spawned in a hatchery, I guess it would technically be the progeny of the fish that are spawned in a hatchery. But maybe more importantly is the natural origin or wild term that really are used interchangeably. And that's the fish that are a progeny of fish that are spawned in nature regardless of parental origin. So that's something important to keep in mind, that some of the goals of these programs are in fact getting hatchery fish that have successfully survived their life cycle, that actually spawn in nature, not necessarily at the hatchery, to promote population conservation. A red is just simply the general location of where female salmon lay their eggs, or some people think of it as a nest as well. And then I'm going to talk a little bit about genetic stock identification. That's where we're a modern method where we're using differences in gene frequencies to identify population or stock origin. So it's actually one of the main ways that we're able to tell when we find a Chinook, let's say in the South Fork, whether or not it came from the North Fork population or the South Fork population. So we're talking about the Nooksack Basin. Here you can see a map. We have the main stem. We have each of the forks: North Fork, Middle Fork, and South Fork. And of course, I'll be focusing mainly on the South Fork, but I will be, providing some information that's sort of relative to the entire basin as general. I'll try and stick to the South Fork, but sometimes it's a little bit unavoidable just because of the way, fish are migrating and moving throughout the entire basin. So thinking about some of the species of concern that we have, of course, we have our Chinook salmon that are within the Puget Sound ESU. We also have pink salmon, chum salmon, coho, and steelhead, which is also ESA listed within the Puget Sound distinct population segment. So I'm going to talk, just give a really brief overview of where each one of these species are at population-wise. So a little brief overview of status and trends. So for pink salmon, this figure is referring to the total basin escapement, so it's not necessarily limited to just the South Fork. We probably could come up with a basin-specific estimate for the South Fork, but we don't have that yet. So that's one of the caveats of these things, is considering, specifically the South Fork, is a lot of these things we think about basin wide. But you can see here the trends. We have odd year pink salmon, I should say. So you can see the years sort of jump every 2 years where you see a data point. And we've seen, you can see fluctuations in the health of the population. We saw a pretty big return last year in 2021. But most of the figures that you're going to see, I only have through roughly about 2019 or 2020, just because there is a little bit of a time lag to kind of get those estimates finalized and agreed to. So I'm just going to use the most recent data available that has been published in some report. Next, we have chum salmon. You can see that again is also a total basin escapement estimate. Numbers have fluctuated quite widely. They started taking a dip starting in 2015, which led to some changes in some of the harvest regulations, and then we've seen a little bit of an upturn in the last few years. Next, we have coho. Coho is a little bit misleading, and I'm not going to go too far into it unless folks have more questions at the end, but this is actually a relative abundance estimate. We don't actually have a total abundance estimate. So this is sort of relative to spawning years that were calculated back in, I think it was the 1980s, the mid 1980s are their baseline comparisons, mostly due to, the lack of capacity to come up with new methods or establish a new protocol. But you can see the coho population is not changing too much. We had a pretty solid return in 2019, and I think as of 2021 we had a good return as well. And so I'm going to focus a little bit more on Chinook. So there's a little bit more in this figure for total escapement. You can see on the left side of the figure, the total escapement numbers are, an order of magnitude lower than the other species, simply because the total population abundance has been so low. So in the figure, what you're looking at is specific to the South Fork population. So the green is the total escapement estimate, and the red is the wild escapement estimate. So you can see in terms of recovery, we're really concerned. We're concerned about both, but we're mainly concerned about that red line to try and get it up to, establish to get, possibly a delisting at some point in the future. So you can see both of those numbers are low. You can see there's a little bit of a difference in the green and the red starting around 2015, 2016. And that is representative of some of those hatchery fish starting to come back, leading to more of that component in the population. So in terms of recovery of the Chinook salmon ESU, they're listed as threatened under ESA in 1999, both populations, the North Middle Fork population and the South Fork population. And so the natural spawner abundance recovery goal under low productivity conditions is 9,900 spawners. And over those last 5 years that I pointed out, starting in 2015, if we look at a 5-year mean, 5-year average, we only have about 300 spawners. So you can see there that we're quite a ways away from our recovery goal. And so, I think we'll get a little bit more into how we're approaching that when we get to the hatchery and restoration sections. So because, the natural spawning population was so low and we're thinking about recovery, that was what led to the establishment of the South Fork Chinook Rescue Program, just a really high level overview of the program. So they utilized juvenile captive brood to start the hatchery component of the population. Those fish were based on genetic criteria. Basically, they tested the fish several times before they even started initial spawning to make sure that they were genetically from the South Fork population, just to make sure they had the best representatives if they were going to establish a hatchery component. And the main goal was simply to increase total escapement for natural spawners, also increasing harvest opportunities. And of course, those hatchery spawners can supplement the natural spawners, the wild fish that already exist in the population. So one of the interesting things about the program is that only a portion of the adult fish return volitionally to the hatchery. So not all of them go back to the hatchery. So you have some fish going into the hatchery that are used as brood for future releases, and you have other hatchery fish that simply return to the spawning grounds and spawn naturally. So just a general overview of what their releases have looked like up through 2019. You can see early on in 2011 and 2012 was when the program was sort of gearing up with those initial releases, and they started ramping it up closer to 2017, 2018, 2019 is the first time they got pretty close to release goals of between 1 and 2 million. juveniles, and the columns on the right are identifying how those hatchery fish are marked. So this gives you an idea of if you see a fish out in the wild, if it is a hatchery fish, these are the ways that in which it would be marked. So most of the fish are coded wire tagged. Recently, we've started instituting thermal otolith marks for fish. This means that we can take out the otoliths and look at them under a microscope and see a specific mark that is representative of which hatchery it was reared in. And then also the most common that folks are familiar with is adipose clipping. So you'll notice that that wasn't commonly used early on because they wanted to get a good idea about what the survival of the fish were in, like, harvest fisheries and natural mortality. But the more of those fish have been getting adipose clipped recently. And so looking at the returns, so are those fish actually making it back to the hatchery? You can see we have some data from 2017 to 2021, and we're getting some pretty consistent returns back to the hatchery, which is good. I mean, you need to have at least a certain number of fish come back so that you have enough eggs and milk from the males, so you can fertilize the eggs. So there's been some good consistency there. And just looking really broadly about its relationship to the number of reds that we've seen, which is representative of spawning activity in the South Fork, you can see since 2000, we've had a pretty consistent number of reds observed in the South Fork Basin, anywhere up, between 100 and 200, a little bit more than that. But since those first batches of hatchery returners started coming in, beyond 2015, we've started seeing a big increase in the number of reds. So there's certainly a lot more involved in that relationship, but it is certainly something to, certainly an interesting observation. So diving now into the monitoring and evaluation side of things, talked a little bit about, escapement and what that number represents. Sort of looking under the hood about what other information we're collecting and what goes into those population assessments. It really comes back to the questions that we're asking ourselves. So of course, one of the most important ones is how many fish can we harvest? How many adults successfully spawned up in the spawning grounds? How many juveniles are produced each year? Those are all the key questions that basically tell us how many fish are there. How many can we harvest without, causing harm to the population? And what's it going to be like next year? What's it going to be like the year after that? So within each one of these questions, how many fish can we harvest? Our co-managers, we conduct annual forecasting. So we put the information that we gather each year into annual forecasting to predict how many adults are going to come back. We use regular in-season catch monitoring and adaptive management. We're always sharing data amongst and between each other to stay up to date and make regulatory changes as needed. How many adults successfully spawned? I'll talk a little bit more about our adult spawning ground surveys, where we actually go out and look. We go out on the spawning grounds and mark reds and find fish carcasses and try and get a good estimate on what's going on there. And one of the newer methods that I'll talk a little bit more about is transgenerational mark recapture. It's another way of estimating adult abundance. And how many juveniles are produced each year will tell us also how successful those adults were at spawning. That will, inform hatchery program management. Let us know, what an ideal number of juveniles to release would be. It also tells us by monitoring the juveniles what their survival is, from the upper river to the lower river. I'll talk a little bit about our juvenile outmigration monitoring program and what that entails. So what data do we collect? This kind of goes between both our juvenile and adult monitoring. Some basic information that you might expect, size of the fish, We can determine sex, not necessarily for the juveniles, but for adults. We get ages for fish through either otolith or scales. I guess I should also say coded wire tags, because that can tell us the age of the hatchery fish. We record mark status, which is, really important when you're tracking the differences between, hatchery fish and wild fish. So we get information on the coded wire tags, whether or not it has an adipose fin, and then we also collect otoliths to read, to get an age or read those thermal marks, which can tell us which hatchery it came from. We collect fin clips. That tells us that genetic stock identification information, and also we use for our transgenerational mark recapture study. Some of the newer things that we're trying to improve tracking is using pit tags to track juvenile survival throughout the river, and also when the adults return, that could give us information on what their marine survival is. We implant jaw tags, which has been interesting. You can see the picture here is a Chinook that we caught this year. Actually, it was jaw tagged in our spring gill net fishery, and the fish was a female, and it was released and jaw tagged, and then we found it on the spawning grounds. So sort of a nice picture of what we hope to achieve by releasing those fish in harvest fisheries. And of course, we collect environmental conditions, and I think that's always the big thing, is how are all these status and trends changing as we look at what the environment looks like at that point in time and what it might be in the future. So talking a little bit about estimating juvenile abundance, we have two main programs that we institute. One is our rotary smolt trapping program that we sample outmigrating juveniles. You can see a picture here is actually an old picture of our rotary smolt trap that we have at Hovander Park that some of you may have seen. We've been operating that since 1994. And so that allows us to sample, outmigrating juveniles and get raw counts. We can calculate abundance estimates. We can collect fin clips and see what population those fish came from. We also do regular beach seining to look at localized abundance and density. We also collect fin clips from fish that we get from beach seining as well. I wanted to talk a little bit about how we estimate juvenile abundance. I might go through this a little quickly because I don't want to get overly technical, but I think it's some interesting information. So the two stars you see here are actually our two smolt traps that we run. The green star is the Hovander Park one in the lower main stem. The red one is a new smolt trap that we just started running in 2021 in the South Fork to help us get higher resolution information on what the juveniles were doing in the South Fork Basin. So basically, you can see, you can assume that where the smolt trap is positioned, we're going to be sampling water that's flowing downstream. So the red smolt trap gives us an opportunity to sample any fish that are spawned or rearing above that, upstream of that. So we know there's some spawning that goes on downstream, but we do have ways to sort of account for that. And then the green trap is going to be sampling fish that are coming from anywhere upstream, which includes not just the South Fork, but the Middle Fork and the North Fork as well. All of the productivity coming from there sort of, comes out, and then we can sample it when they get to the lower river. So we're trying to put these two things together to see what the spawning success of the parents are, what the juvenile outmigration timing and abundance are, and get a little bit better idea of what the survival is in river. I want to share some of these results with you because I think they're interesting. So first, I'm going to show you, this is our main stem smolt trap. So this is the lower river. This chart is showing the periodicity of our catch throughout the year. And I think it sort of clearly shows some trends that you can see, where earlier in the year, you can see Statistical week 8, that's about, the end of February. And then the y-axis is proportion. So it's just saying the proportion of total catch within those weeks. You can see early in the year, we're pretty much only catching chum. We're catching those chum fry, those sub-yearlings. We get a little burst of coho. That's mainly from the hatchery. We're catching mainly those yearling coho that are raised in the hatchery. We don't get many sub-yearling coho in the lower main stem trap. And then you can see later in the season, we get mostly Chinook. And that includes both wild Chinook and those hatchery releases. So if we look at the catch per set, this is focusing only on Chinook. I wanted to just highlight the difference between the hatchery Chinook, which are in black, and the wild Chinook, which are in red. And you can see there's actually some similarity between the two. You can see a couple of blips where you get higher catches of both wild and hatchery. But also take a look at the axes. We're catching about 5-to-1 more hatchery fish than we are wild fish, which certainly isn't unexpected. But it's also interesting that we're not really seeing a big difference in timing. We're kind of catching those hatchery fish at similar times that we're catching the wild fish. And if we look at those same figures on our South Fork smolt trap, we see a little bit of a different story. We didn't fish it as long, so you can see the statistical weeks don't start until late April. But we were catching those coho early in the season. Were actually sub-yearling coho, so we're catching those age 0 coho, which was interesting. And we're catching some wild Chinook earlier in the season. Then you can see that big boom in coho catch. That's representative of the hatchery fish. And then also we have more Chinook being caught a little bit later. Also some hatchery fish. What was cool too is this the bunch of steelhead that we got later in the year. These are actually age 0 steelhead, which we don't really catch age 0 steelhead down the lower river. So we're starting to get some information on those fish that we're not really seeing otherwise. And if we look at our Chinook catch per set, again, we see a pretty similar story. We're looking at the black is the hatchery fish, the red are the wild fish. You see a pretty similar trend as far as when you're being caught, and we're still catching, more hatchery fish than we are wild fish. So this is just a good example of some of the information that we're using to kind of track, the differences over time. Switching to adult abundance. So now we're tracking the adults, and the main method we use that for those are spawning ground surveys. You can see in the top picture, it's a good example of what our recent days and weeks have been like in the upper river, where we fish as many carcasses out of the river as we can, and we process those. The bottom picture shows you an example of a live fish in the water next to a red. We count all red and identify them and mark them. And also we do this across the basin. So as I said before, some of the information is relative to the entire basin, though I'll try to give you information that's specific to the South Fork, especially when it comes to the restoration stuff. So again, we're looking back at this, escapement figure. The green is the total escapement. The red is the wild escapement for the South Fork population. So in the South Fork, we have a red-based abundance estimate. So actually the way that we calculate this green line is not dependent on the number of carcasses that we find. It's completely dependent on the number of reds of salmon reds that we identify and mark. So it's a red-based abundance estimate, and we identify all reds through October 8th, which according, I think, is almost 2 days from now. Yeah. So our team is out there, not as we speak. I think they just got out a little bit earlier, but they're out there marking all their reds right now, and that'll go into the abundance estimate. But the way that we actually come up with the partitioned abundance estimates is by using those carcass recoveries to look at the composition. So we use carcass recoveries to tell us how many North Fork fish are there, how many South Fork fish are there, how many hatchery fish are there, how many wild fish are there. And so we are exploring new methods, and one of those is transgenerational mark recapture that I'll briefly touch upon. So that, when you look at the figure on the left, the gray line is sort of our existing red or carcass estimate. The North Fork population uses carcass count, so that's why that is there. The TGMR is a new method where basically you can collect DNA from your adults and your juveniles. You can know how many adults you've sampled because you have the complete census data from the spawning ground surveys. You know the amount of fish that you've collected DNA from. So basically those become your marks. And when you recapture the juveniles, you will get a parental assignment if they are genetically related to a parent that you have previously sampled. So it's sort of a new and novel way that's been used. In fact, in some other watersheds, I think they've actually switched from older methods to a TGMR escapement method that is used in harvest planning and population research. So you can see a little bit of the comparison, and this is for the Nooksack watershed. There's certainly a few caveats to it, a few assumptions that are being made, but you can see there are definitely differences in the abundance estimate based on the method that you're using. I have a note in there to say that this figure represents both Nooksack Chinook populations combined. It's not specific to the South Fork. That's mainly because our lower main stem trap samples from both populations. So I thought it would be interesting to share some of the new methods that we're exploring to improve not only our abundance estimate, but really you can see the uncertainty that's included around each of those abundance estimates. So that can tell us a little bit more about how good are we doing at estimating that total abundance. So that's a pretty broad overview. And now I'm going to talk a little bit about the restoration program. And before I dive in, I know there's some restoration folks on the line here. So I'm going to do my best to talk about what other folks have talked about. If I gloss over something or I misspeak, definitely dive in the discussion portion. But certainly I'm not necessarily the the person in charge for a lot of these restoration actions, but I definitely have gotten some assistance from some of the biologists and restoration managers on some of the content included here. So some of the restoration goals and strategies, I said in the beginning, we have, restore self-sustaining conditions in Nooksack Chinook habitats. You can see the picture on the right there, historical logging practices, just the general practices of land management have certainly led to some of the degraded habitat conditions we have today. And so, pulling some of the actions right from our, salmon recovery plan that we've written, we have, the planning of restoration projects, we have analyzing the impacts, using those population monitoring information as well, land acquisition, actually implementing those restoration projects, the CREP program implementation, and forest land management. And really the question is, what factors are limiting recovery in terms of what are these restoration projects hoping to achieve? So some of the key limiting factors are a low habitat diversity, a lack of key habitat quantity. Specific to the South Fork, we have high water temperatures related to low flows. Some of the products of those, the degraded habitat, are disconnected floodplains. We have a lot of red scouring of spawning beds that occurs, and there's been a few research projects that have sort of quantified that and shown that that is a big problem. lack of refugia for fish, including, primary pools for those fish to hold in during migration, and reduced channel and edge, complex habitat. So I want to talk a little bit, sort of focus on log jams as an important recovery strategy. And that's been true across the basin. You can see in the North and Middle Forks, they've been dealing with some, channel instability issues, low habitat diversity. In the South Fork, high temperatures, low flows, lack of pools, and log jams have been an important recovery strategy in both forks. And you can see all of those dots represent, log jams that have been implemented in the South Fork basin over time. And over here, the figure on the right shows the total number of log jams that have been constructed. And so completed, there have been 18.3 miles of habitat that have been completed, but you can see, the number planned, 53.7, is still quite a high number. So despite all of the work that has been going on, there's still a lot left to be done, especially when you consider relative to that recovery goal for Chinook of 9,900 spawners, 9,900. So this figure again kind of shows the cumulative effort between some of our co-managers and constructing log jams. I also note the Whatcom Land Trust has acquired over 3,000 acres, and land acquisition and preserving land is definitely an important action as well. Lummi Nation has focused a bit more on the upper South Fork, constructed 156 log jams. Nooksack Tribe focusing a bit on the lower South Fork, constructed 95 log jams. And over $16 million in surfboard and PSAR funding have gone to these efforts, but certainly a lot more is needed. Just going to give one example. This figure shows Lummi Nation restoration projects within the South Fork, and there's a little bit in the Middle Fork too. I just wanted to highlight one of the projects to talk a little bit more about it. Certainly there's a lot to choose from. And down here around River Mile 20, we have the Larson's Reach. And so in this area, the goals were to reconnect floodplain, combat channel incision that was going on, slow the river, slow floods, and reduce the transport capacity, and also, upgrade the channel overall. You can see here, this is a picture from 2018, post construction of a pool that was formed. This is photos from Ian Smith. You can see a bunch of different species, nice sockeye there in the front, some coho and Chinook that are holding in the pools that were formed. This figure shows sort of the before and after. You can see the green is the before, the blue is after. You can see the big change in the number of pools, relative area in the restoration area really changed. So the number of pools went from 9 to 37. So a big increase in that key habitat that those Chinook need when they're migrating upriver. And this figure shows the number of reds in that project area. So again, the big takeaway is you can see over time, going all the way back to the 80s and 90s, you can see how the reds were sort of spread throughout this project area. But the mean number that we saw pre-restoration and post-restoration dramatically increased, almost doubled, or more than doubled. So that's a nice effect that we might be able to at least partially attribute to the restoration project. And so some of the overall observations that we've seen from these restoration project implementation is an overall increase in pool quantity and quality, increase in habitat diversity, increase in side channel length, and increase in forested island counts. An estimate was that 93% of Chinook were holding in these wood formed pools, which is an important part of their life cycle. And overall, there was a recent assessment that, we're partially effective at meeting our overall goals. And I would say the projects themselves are certainly effective, but partially effective being still relative to our total goals of, the miles that need to be restored and meeting those population recovery goals. We still need to actually implement more projects to get those to where they need to be. And I just want to finish a bit with the broader concerns that leads us to, in terms of, not yet meeting recovery goals. Certainly folks are aware of, sort of the double whammy that we got last year in 2021, where we had, a heat wave and high water temperatures between June and September. The picture on the bottom is from a pool below the Skookum hatchery, where you can see some of those white dots. Those are some fish that didn't survive, weren't able to make it all the way back to the spawning grounds. And then, of course, the top picture, we had, major flooding events in November 2021. Even the fish that were able to spawn in the summer, their reds can be impacted by flooding events that might very well, wash away any larvae or juveniles that are rearing at that time. And just as an example to give you, this is pure raw count. So this is from our lower main stem smolt trap in 2022. So 2021, we had, the heat wave and the flooding, which is represented by that red line. And you can see 2022, I just wanted to show you what our preliminary counts are of wild Chinook. You can see it took a pretty big dip. So I don't have, an official abundance estimate for you, but I think it was clear to our team that there definitely was some impact to, either or both of those situations that occurred. So again, definitely a high level overview, and I wanted to just include a slide to show, for more information, go to the Lummi website. There's lots of information there. The Wire One website has a lot of information from the salmon staff team and all of these, the recovery plan and restoration projects and actions that have been going on. The Puget Sound Partnership has some good information too, both locally and regional information. And I'm happy to share my email if anybody has specific questions to follow up with as well. And just thanks to the Lummi tribe and the Lummi Natural Resources staff who have helped out both with the projects and with the presentation, and of course the Nooksack Basin co-managers for operations and support. And this is all tied to a whole lot of grant funds that I don't think is certainly exhaustive, but just an example of a few of the grant sources that we get. And with that, I'd be happy to answer any questions.
Great. We have just a few things in the chat, and while you answer that, folks can think about whether they have additional questions that they want to ask that they can put in the chat or raise their hand, I guess. The first question, I guess going back, people kind of answered, I guess, Kim, if you have any additional questions about what escapement means, please let us know. Otherwise, her next question was, is Larson's upstream of Kavanaugh Creek?
Yes, it is.
Thank you. And then Colin asked, to what extent is estuary habitat a limiting factor for Chinook recovery in the Nooksack?
That's a good question. I don't have a simple answer for you. There were some studies. There was a study done a few years ago that basically showed there was, some density dependent mortality going on in the estuary, but that it wasn't necessarily being limiting for the Nooksack populations. I think it's a question that still we sort of want to get more information about, but I think there's a difference between looking at the estuary as a place that we should think about as a restoration project to like restore function, but it doesn't appear to be a limiting factor in, in population productivity as far as I'm aware.
Thank you. Lindsay just had a clarification that the number of log jams constructed by the Nooksack tribe was specifically just for the South Fork, all of the lower South Fork downstream of Saxon, of the Saxon Bridge. The Nooksack has constructed over 260 log jams in the North Fork Nooksack. And then Kim asked, could you describe the restoration plans for the Whatcom Land Trust property just south of Acme?
I could not.
I could probably help with that, Devin.
Okay, go ahead.
I think Kim, you might be referring to the property, yeah, on the west side of the South Fork, across from the Whatcom County South Fork Park, if that's correct. We have a project called, Nooksack Tribe has a project called the Fish Camp Project that's being planned out there. It's a collaboration with Whatcom County and the tribe to develop design and a project to reduce flooding in Acme, but also restore salmon habitat in the main stem South Fork Nooksack. We have a project website. I can put it in the chat box, and there's all sorts of information on that project webpage.
Thank you. Tammy asked, what is happening to the South Fork Chinook right now with the late summer drought?
Yeah, good question. So let's see. It's kind of interesting out there right now. I guess I should start off by saying we're actually seeing sort of a record spawning activity going on right now in the South Fork. I think and this is just based on raw observations and raw data so far, but I think our team has marked more reds than we have ever since your recording has began. So that's a good place to be. More spawning activity than we've seen in a long time. It's a good sign for the population. I would say we did have some pre-spawn mortality, some fish that never made it all the way up to the spawning grounds, presumably due to high temperatures, low flows, prevalence of bacteria. But focusing on the former at this point, yeah, we're seeing more fish. We've worked through more carcasses than we ever have. And so the spawning seems to be really strong this year. We're hoping that, of course, we're all waiting for the rain and the river to rise up a little bit, but we're also hoping we don't get a repeat event of last year, which could very well, really impact the ability of those reds to produce offspring if the floodwaters, impact that too much. So we'll have to wait and see. This is sort of where the cycle goes. We'll get to our juvenile monitoring next year, and then that'll tell us really, how successful the adults were at spawning.
Thank you. Brittany asked, since the chum populations seem to be dropping, are there any management tactics going to be put in place to bring those populations up?
That's a good question, and I'm not as involved on the management side of things. I'm sort of more of the person that gives the relevant information to the managers to decide. I know there's discussions about, the hatchery programs that are going on and making some adaptive changes to those to bolster populations. And I know that, harvest regulations are always, on the table to see how those might be impacting, the fish coming back. I don't really have any concrete answer for you, but I can tell you that it is a hot topic, and folks are talking more about chum these days than they have in a while.
And I have a question that I feel like I try to always ask our speakers. Are there any ways that you can think of that the Whatcom Marine Resources Committee could plug in and help support efforts like this?
That's a good question. Well, I think the first thing is, giving this presentation was really nice because I feel like, you know, the co-managers do a lot of work, but sometimes, we're a little bit disconnected in some ways, just because it does take a lot of effort to, carry out these projects and these surveys. And we are always talking about, outreach and getting involved. And a lot of this stuff we don't intend to, hide from people or anything like that. It's just we don't necessarily have the time to share it all. So yeah, I think this opportunity is great. And again, I encourage anyone who has more questions, to reach out to us, check out those, websites and sources. A lot of times we're kind of just citing that information that might otherwise be available somewhere else. So yeah, thanks for the opportunity, and we'll hope to do it again, and maybe update you on some of the efforts that I talked about that are ongoing.
That would be great. Yeah. Lindsay, who works for the Nooksack tribe, who is also part of the whole co-management thing, said that feel free to reach out if anyone has any questions. They just had their 3rd community workshop in Acme a few weeks ago, and we'll be updating the webpage soon with materials. They will present at that workshop. And she also added, this is a great overview, and thank you, Devin. And I also echo that thanks. And others are also now saying thank you. Are there any last questions for Devin? Going once,